Yes, I know I promised to put the first quiz up, but I didn't do it yet. I will shift to the first quiz for this week, so don't panic. You'll get it, all right? Oh, I actually printed an extra copy of lecture 3. I forgot I had printed before, so does anybody want colorful images to use? All right, it's auction. Who? Oh, okay, fine. Will you pass it to me back? Okay, so let's dive now into more detailed mechanisms and molecules, and again, things that you have to know because that's what people taking immunology have to know. Okay. So here is really going to be a deeper explanation on mechanisms and molecules that are important for innate immune system, so innate immune activity. This here is for your practice, so this is not part of this iClicker thing. It will take half an hour to answer this, so that would be like half of the class. But here we start with points for you. So let's get your iClickers going. And this is just to make sure that you have watched the videos and you know what the cells are doing. And this is the time when you can totally cheat and ask your neighbors and make sure you get the right answers. So I'm starting here the iClicker. And basically the question is, when you see eosinophils, what does come to your mind? What did it come to your mind, right? Somebody come in, rush, you get time to get your first 2 points. iClicker. All right. So when you look at eosinophils, what is the first thing that comes to your mind? Does anyone need more time? Yes, all right, give you a few more seconds. People know. If I don't remember them, you don't have to. Okay, so everybody answered? Okay, I'm stopping it. Should we give him a chance to hop in? Do you want to get iClicker points? Yeah, I give you 15 seconds. That's a long time. We're going to 2 minutes now. Now, big clue in the answer here. Oh, good. Everybody shifting to the correct, most everybody shifted to the correct answer. All right, you got it? You got the points? Okay, so here we go. We got over 2 minutes for this answer. What's your answer? C. Why isn't it A? Oh. Why isn't it A? They are rare in circulation. I don't care if they stay in blue or pink, that's not my problem. Okay. Yeah, the A, this says non-phagocytic. And eosinophils, they are not major phagocytic cells, but they can phagocytize if they want to, okay? So here, the answer really, the biggest clue for this answer is telling you that it's defending against parasitic worms. When you see parasitic worms, you think eosinophils. Also, when you see allergies, you think eosinophils, right? But mainly here, against parasitic worms. So that's a big clue for your answer. Now, which one is the cell that is most abundant of the leukocytes? Start with an N. Neutrophils, yes, those are the most abundant in the circulation. Actually, most abundant. Okay, so now we are going to this specific, objectives here, that we're going to look more into these mechanisms, and we are going to look a lot more into these pattern recognition receptors, and we're going to know how they work and what they are used for. So we're going to have to know some characteristics of those, where they are located, and what they are meant to attack. Then we're going to look at some mechanisms of activation of inflammation. Basically, we're going to look at NF-kappa B activation, and we're going to look at this cool one that is NLRP3. I know, alphabet soup, right? But you're going to think about inflammasome, NLRP3, and all the stuff that's involved with it, okay? So two very important mechanisms, and then location and function of these molecules. And this you're going to find in chapter 4. And don't get scared when you read the book, because it's a lot of details in this book. There are a lot of PRRs and lots of targets, and oh my goodness, every time I go back to the book, I'm thinking, okay, so we'll focus on what is, yes, they take forever. And then once you start reading them, if you're not fully awake, don't drink too much coffee, by the way, it's not good for your heart, right? If you're not fully awake and you start reading and you go, blah, 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 like Charlie Brown listening, Charlie Brown classroom, right? You're trying to read blah, 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 blah. Yeah, sorry. But so I take some things from the book, some other things from other sources that are a bit more exciting, right? So here, again, remember that your innate immune response does not keep a memory of the thing that it attacked, although that's not 100% correct, but for the purpose of learning what it's doing, so it doesn't have a memory, okay? It attacks the things exactly the same way that it had attacked the first time. It doesn't mount a better and faster secondary response, so that's the job of adaptive immune system. And remember, we are looking at layers, right? So the first layer of protection, you have, the surface barriers, right? You have the mucus, so you have the physical barriers, so that involves your skin, that involves the little hair that is coating the mucous membranes, right? Cilia, right? So those little hairs that beat and push things out, like for example, of the airways, those are cilia, right? Then you also have, okay, here's showing the mucus. So those are all physical barriers, right? And besides the physical barriers, you also have those chemical barriers, right? So you have some enzymes that are very important to break wannabe invaders. And you have some things like acidity. So low pH in your stomach is also important for destroying wannabe pathogens or invaders, okay? So all first line of response. And this is when I tell this story of how much do we rely on our innate immune system, right? Because most of the pathogens that get to you, they don't survive. They never pass these first layers of your innate immune system, right? So my example of this is when my older son was about 1 year old or so, right? And I had him when I was in my first year in graduate school. I recommend nobody to do the same if you want to survive and still keep your sanity, right? Baby, graduate school, that's a bit insane, right? But yeah, I did it. I survived. And after I survived, like 5 other women in the graduate program decided to have babies too, and the department did not like me very much. Anyway, I think it was something in the water. Okay, so back to this story. What happened, right? So what happened? I'm driving my baby to school. I'm going to UM, you know, so he had to go to the daycare right in front of UM. There's like UM Canterbury. So I'm rushing every time, get him to the car, get to school, get to work. And then I am, you know, on 95, and I look at the mirror, and I see he picked up something from the seat that looked like, I don't know, a cookie, chocolate chip cookie, a piece of brownie. It actually didn't look like that. It had like green stuff growing in it. And by the time I realized that he's gonna, of course, what do babies do? Everything goes to the mouth, right? By the time I realize he's gonna put the thing in his mouth, I'm like, 95, cars, crazy people, bye bye. Like, oh, too late, it's gone. I'm like, ugh, what am I gonna do now? I'm not gonna pull out a 95 and try to make him throw up. Bad idea. And then I'm thinking, oh, I know about the immune system. Innate immune system protection. So he has the mucus, he has the low pH in the stomach. That should definitely get rid of whatever was growing in that piece of whatever it was, right? And then I'm thinking, I just hope he doesn't develop a fever. Please don't develop a fever. And you can imagine I didn't sleep for about 2 days because I was just so afraid I was going to develop a fever. And that would be the next stage of innate immune attack, right? But then I was really hoping that he was going to stay on the first stage, like whatever tried to get into the body, we don't even know about it because the innate immune system is so efficient at killing the stuff, right? And sure enough, he did not develop a fever. I was like, oh, thank goodness. Okay, thank you, the barriers that really came into effect. So it was actually very effective for killing, I think, of course, was some kind of fungus, right? Whatever that was. So he survived, no problem. Innate immune system jumped into action. Adaptive immune system never had to be recruited. Okay, so what happened here? You have these barriers, so we have to know what they are. And here I like this figure from the book because it's really putting into perspective. Look at this. You have acidic pH, okay? You have antimicrobial peptides, which there are a few of them that you have to know. Okay, so get ready, right? You have the mucus, all right? You have physical, mechanical barriers. Now, let's say that thing will go through this physical barrier and chemical barrier. Now, you're going to have to start recruiting your cells, right? So depending on whatever came across, you're going to have an app for that or a cell for that, okay? So let's see here. You're going to have some phagocytic cells. So here we have neutrophils or tissue-resident macrophages. Remember, there are different types of macrophages, right? You got to know those, right? So some macrophages here that can try to eat up the pathogen, and they can start secreting stuff, okay? Either antimicrobial peptides or signaling molecules that are going to further activate the ability of the cells to phagocyte, to perform phagocytosis, right? Now, these molecules here, these chemokines or cytokines, they are also going to work in the environment around this invasion over here. They are going to work in the blood vessels. Okay, and they're going to start causing inflammation. They're going to start causing specific reactions that you're going to see in the blood vessels, right? Now, all of this, okay, of course, besides your myeloid lineage innate cells, you also have the lymphoid lineage natural killer cells that, again, depending on the pathogens, they might jump into action and try to kill the pathogen, okay? So all these things that are happening here, this is all part of this mechanism of your innate system. And then hopefully we stay around here, meaning the pathogen is killed and nothing happens afterwards and we're all happy, okay? But it could happen that you would need to activate your adaptive immune system, and then that activation is going to happen in a secondary lymphoid organ, okay? And the cells that are going to be responsible for the activation, those are going to be your dendritic cells, mainly your dendritic cells. Then, of course, we're going to see more on that activation later. Okay, so all this part that is happening right here in the beginning of all these responses, those are all your innate system that came into action. All right. So what are the microbial, antimicrobial peptides and where you're going to find them? Okay. Okay, so hopefully oh, this thing is not working anyway. Can you hear me? Can anybody hear me? Yeah, in the back. Is this thing working? All right. I'll just talk loud. So where are you going to find these antimicrobial proteins? And then the acronym is AMPs, right? So antimicrobial proteins, you're going to find them on your skin and other epithelial layers, okay? And examples of AMPs, you have lysozyme. So that's your first enzyme here. So lysozyme is going to break something. It's going to lyse something, okay? What is it going to lyse? Well, mainly it's going to look for stuff that is in bacteria that we don't have, like, for example, cell walls, right? Our cells do not have walls. Bacteria, they do. So they're going to break bonds between sugars in the cell walls, okay? So the bonds they're breaking really nam and nag. No, I'm not going to ask in specifics about the sugars, but it really is breaking sugars in the cell walls, okay? Look how many of those you have. 22,000 amps that can break stuff in bacteria, okay? And have other functions as well, but it's a lot of antimicrobial peptides, okay? All right, so sources and functions, where do you find them? You find them on your skin, you find in tears, in the saliva, milk, you find it in your intestines, okay? And of course the skin here. All right, so what is the mechanism that these AMPs are using? Some of them just go and cut directly the sugars in the cell wall, right? And that's antibacterial as antifungal action. Some of these AMPs, they grab iron. And without iron, a lot of bacteria can't really grow. So that's another strategy, okay? And some AMPs, instead of disrupting the cell wall, they can disrupt directly the bacterial membranes, okay? So that's still another mode of action, okay? Besides all that, some of those, they can just bind the surface of bacteria and prevent that bacterium from binding to the cells that they want to infect. Okay, so it's like a buffering or coating of those microorganisms. Okay. And this is really interesting here. Look, besides your own body producing these AMPs, you also have your good bacteria, your commensal bacteria in your intestines that are generating some of these AMPs. And that's really neat because the good bacteria are producing antimicrobials, meaning a lot of them are just antibiotic, that are going to kill the bad bacteria. Okay, like what kind of antibiotic? So we have here, for example, penicillin, polymyxin B, vancomycin, that's a good one, okay? So your good bacteria are helping you to fight off the bad guys, okay? Now, a lot of the AMPs, they can be used in several ways, like you kill several things, and a lot of them are redundant. So if one is not working, or another one can pick up the job of the first one, okay? So if you have a good diet and you really help your good bacteria, then you're going to have this extra layer of protection against these potential pathogens or opportunistic pathogens. All right, so things that you have to know, some peptides that you have to know. First one is your lysozyme, okay? That is going to cut the sugars, mainly peptidoglycans, in your cell wall. Okay, and then the bacteria is going to burst because it doesn't have enough pressure to keep the stuff inside. So kills the bacteria. Then you have this guy here, look, lactoferrin. When you see ferrin, you should think iron. Okay, now where you're going to find this? You're going to find it in mucosa, okay, in secretions. Okay, and that is what is going to bind iron. Okay, so that binds iron and sequesters. It binds iron and eats it all up, so bacteria can't have it. So the bacteria is not going to grow. Okay, so we have here these two examples. Now let me see what else. Oh, and then you have this guy here, APO3. This is in the intestine. Okay, this is interesting because it is going to disrupt the membrane of certain types of bacteria, like gram-negative bacteria. Okay, so gram-negative bacteria, they don't have cell wall, but they have double membrane. Okay, so if you don't have cell wall, then lysozyme is not going to work against it. But then this other enzyme here, they're able to break down the membrane of this gram-negative bacteria. Okay, so this is the outer membrane of your gram-negative bacteria. All right, so these three here are important for you to know. Okay, and then you have some more here, like for example, defenses. Defenses are going to be on your skin and respiratory tract. They are also going to disrupt membranes, okay? But these are going to be bacteria, fungus, and other parasites, and they are also going to disrupt viruses. Okay, so defenses, they are working against everything. Where you're going to find them? On the skin and respiratory mucosa, or also in the intestine. Okay, skin, mouth, mucosal tissue, mucosal layers. All right. And look at these guys here. His statins. Okay, his statins. Those you're going to find in the saliva. Okay, what they're going to do? They're going to bind to certain cell walls that are characteristics of fungus, okay, fungi. Okay, so his statins are going to kill the fungi. Okay, then you have this other type here that you have here, dermicidin. I got to check on the spelling of that. Okay, that one is interesting because you find it on the skin. Basically, it's being produced by your sweat glands, and that is also going to attack membranes of pathogens. And look what it does here. This thing is antibacterial and it is antifungal. The way that this dermicidin works is by punching holes on the membranes of those pathogens. Okay, so if you punch holes, you're going to disrupt a lot of gradients inside of those pathogens, and that's going to cause their death. Okay. And this one is easy to know that it's in the skin because derm, and anything like derm is basically, you know, dermatology, right? It is on the skin. Okay. All right, so what is happening so far and where are we? So innate immune system, they have to recognize what they have to attack. They have to activate some cells and some other pathways. They have to produce some kind of signaling molecules, right? So signaling molecules are going to be cytokines, but they're also going to be responsible for inflammation. So they're going to produce proteins that are going to cause acute inflammation, right? Besides all that, those same cytokines, they are going to upregulate the stickiness of some certain cells. So they're going to increase the production of cell adhesion molecules that is going to be both in the cell that is jumping into action, but also in cells of the endothelium of your blood vessels. Okay, in which, of course, we're going to look more at it later. But if you have here your blood vessels, okay, and inside of the blood vessel you have the endothelium. If you start to upregulate molecules here, you're going to be able to attract certain cells to go over there and interact with them. Okay, so upregulating adhesion molecules is very important, not only for the cells that are they're the effector cells, but also for cells in blood vessels and in tissues. Then those cells are going to have to recruit cells, other cells, to come and help. So the upregulation of these adhesion molecules is going to help also with that recruitment, right? After all that, when you have all your soldiers in place and everybody is active, you're going to have to kill the pathogen. You get rid of the stimulus. So once you don't have pathogen, then your cells have nothing to do, right? So they are going to be bored to death, and they're going to reduce in numbers. You're going to reduce your response, right? Minimizing your immune system reaction here. Then after all that, then your body realizes that your immune system, your innate immune system did some damage to some of your own tissues, so you have to do some tissue repair. So this whole thing is the overview process of the innate immune response. So let me see here. Oh, this is a very important slide that, of course, I don't like to be reading the slides, but I put it here so you can go back to it and study. Okay, look at this. A main strategy of the innate immune system is to block specific things that are needed for the microbes to survive. Okay. Okay, let me see. Okay, so let me see where this is here. So at the same time, the your innate immune system is trying to attack molecules that are crucial for the survival of the pathogens. Some pathogens are very smart, and they really come up with defense mechanisms that are going to counterattack that initial strategy. Okay, so it's always this this type of who is going to win in this battle type of mentality. Okay, so if the pathogen can figure out a way to overcome that initial defense, then of course the pathogen is gonna survive. Okay, and again we're gonna see specific mechanisms of how they do that. Here we have another question, kind of lengthy question. Well, so we are talking really about the innate immune system. Now, which one of this would be a response of your adaptive immune system? That's an easy one. Talking about adaptive here, not innate. Which one is characteristic of adaptive? Okay. Anybody need more time? Okay, I'm stopping it. What's your answer? D. Those are going to be your B cells. Remember, those are your lymphoid lineage cells. And those are going to specifically produce antibodies. So this has to do with adaptive system, not innate, right? Antibodies are very, very specific. All right, so myeloid cells that you have seen this before, hopefully you watched the movie, right? What they're going to do, they are going to identify and try to kill the pathogen. They're going to ring the alarm bell and alert other cells that something is happening. So what are the cells that are used here? All of those cells of your myeloid lineage. Okay, myeloid lineage. And you also have these lymphoid cells that are specific for your innate system. So these are your natural killer cells. Okay? And there are two forms of your innate cells. Okay? Natural killer cells and innate lymphoid cells. So yeah, it kind of sounds like the same, but they're not. Okay, they work more or less similar ways, but they have certain differences. Now, those guys, yeah, they are lymphoid lineage, right? But they have on their surface specific pattern recognition receptors, right? And that is a characteristic of your innate immune system. what are they going to recognize? They're going to recognize pathogens as well as damaged cells or even stressed out cells. Okay. Now, look at this. Main differences between natural killer cells and innate lymphoid cells. The natural killer cells, they like to swim around in the blood. So they circulate. You're going to find them in your blood. Okay. The other ones, the ILCs, they like to sit in tissues. Okay. So they like to be resident in tissues. All right. So main differences between these two. All right. Now, when you look at your innate lymphoid cells, so those are the ones that are sitting in the tissues, right? Innate and lymphoid lineage. They are specialized in attacking different types of pathogens in different ways. Okay, so you have mainly three types of innate lymphoid cells. So let's see what happens here. When you have the first type here, type 1, those are going to look out for bacteria, but not any type of bacteria, only the bacteria that can go inside of the cells. So type 1 is going to look for intracellular bacteria. And how are you going to remember that? So you remember that intracellular begins with I, and I in Roman numerals, that is type 1. Cool? Now you have to think that that's intracellular bacteria. Okay? So intracellular bacteria. So if you look here at the type of immune response that you're having, type 1 is going to be mediated by your first ILC type 1, okay? And that is going to fight bacterial infections, okay? But this slide, this diagram I put here to give you the bigger picture of the reactions as well as of the ILC type. Okay, and this goes very much hand in hand. Although the type 1 reaction of defense could also include extracellular bacteria. But let's focus here on the ILCs, okay? So type 1 is going to fight bacteria, is going to produce a type of immune reaction that is specialized in fighting bacteria. Then you're going to have your type 2 cells, okay? Those type 2 cells, what are these cells again? What am I talking about? They come from the lymphoid lineage, right? And they are innate immune cells. Kind of a natural killer, but those are lymphoid innate cells, okay? Now look at this. These type 2 cells, what is it? Do you remember what the lymphoid lineage cell looks like? They basically look like this. Huge nucleus, very little cytoplasm. Those lymphoid lineage cells, they look like that. If you just look at them, you can tell them apart, okay? So these guys here, let's say this type 2, they are going to be very important at stabilizing or establishing a type of immune reaction that is going to be specific against worms, okay? So you have here against helminth infections, but when you don't have enough worms to stimulate your immune system, those guys also go crazy and can cause you some kind of allergies, okay? So they are responsible for allergic responses, and they are also responsible for activating those eosinophils and other cells that are important for against worm infections, right? So that's type 2, all right? Then you have a third type of immune reaction that is called type 3, okay? So those are going to be specific against, mainly against fungi, and then some viruses and some extracellular bacteria, okay? So here you have this overall picture of 3 types of immune responses based on innate immune system and based on the innate lymphoid cells of the immune system, okay? So type 1, you always think bacterial infections. Type 2, you think worms. Type 3, you think more of fungal infections and or some extracellular type of pathogen. Okay. So this is just to reiterate what I just said before. major type of immune responses are going hand in hand with the types of ILCs. There we go. Inflammation, major function, protective function of innate immune system. And what is the other one? Antiviral defense. So you absolutely have to know that. Inflammation and antiviral defense, okay? What is going to cause inflammation? Well, you have some cells that are very good at that, especially your neutrophils, okay? So you have your circulating white blood cells that are going to be very good at causing inflammation. And you also have a lot of plasma proteins that are produced by your liver, okay? Those proteins are also going to help to produce inflammation, okay? Right? Now, for your antiviral defense, this is really, really interesting because this kind of response is going to happen from the inside of the cells. Okay, so this is a type of response that all cells have, all nucleated cells have, and they can start producing certain cytokines that are going to help themselves to produce antiviral proteins, and they're also going to help their neighbors to ramp up their defenses, alerting them that there is some virus around. Okay, so you have antiviral defense, and the most important molecule for antiviral defense is really your interferon type of molecules. Okay, and again, we're going to see more of this later. So inflammation and antiviral defense. All right. All right, so this you can read just to emphasize those things we talked before. All right, so we're passing through the layers, right? Pass through the physical, the chemical, and now we look at the cells, right? You're also seeing the cells and their personalities and what their nuclei look like, right? And now we are going to get into the mechanism of how these cells are working. And then on the next lecture, we are going to look at the complement system. All right, so now let's look at this pattern recognition receptor. So this is the cellular layer of defense. Okay, where are you going to find them? Well, you're going to find them in several cell surfaces. And these patterns, they are going to be, these receptors, they're going to be, you know, exactly the same thing on different cell surfaces. Well, also, you're going to find PAMPs or DAMPs that also look the same in different pathogen surfaces. Okay, so here you have, for example, a cell that can infect, let's say this thing is one type of bacteria that has a certain PAMP, and here it would be like another type of bacteria that has the same type of PAMP. Okay, so you're going to have this, you can have the same type of pattern of pathogen in different types of pathogens. Okay. We've seen that before. Okay, all right, we see that. Okay, so this is when the fun starts. What are those PRRs? So you have a lot of classes of PRRs, several families of PRRs. And don't worry, I'm not going to ask you just to know all of them because that's pure torture. It depends on how mean I feel certain times. No, I'm not that mean. Okay, so certain types you have to know or familiar with. First one is the toll-like receptor. They call it toll-like because there is certain molecule in a fruit fly that is called toll, and I don't know, I don't care what they call it. So toll-like receptors. Then you have another one, it's called C-type lectin receptors. Then you have Rig-I receptors. Then you have NOD receptors and also AIM type of receptors. So you got to be familiar on what is it that they are doing. Okay, so toll-like receptors, very, very, very important. They will enable you to recognize pathogens that are outside of the cell in the environment, and then pathogens that try to get inside the cytoplasm, and then even pathogens that get phagocytosed and are inside vesicles. You got like a toll-like receptor for that. So there are many, many of those. Now, C-type lectin receptors, those are going to be important for recognizing certain patterns of sugars that are in the cytoplasm. Of course, of these pathogens that got into the cytoplasm. When you look at RIG-I receptors, those are important against viruses, okay, viruses. Then when you look at NOD-like receptors, those are very important for intestinal mucosa defense, okay? And the AIM2-like receptors, I don't remember them now. So which ones are the most important? Those guys over here, okay? Here, what is it that this is straight from your book, okay? So it's telling you basically that if you have extracellular pathogens, you have a receptor that is going to recognize them. If the thing got phagocytosed, you're going to have activation of another receptor that is going to help you kill it. If the thing got in the cytoplasm, you're going to have a receptor for that. So anywhere this pathogen wants to attack the cell, is going to be met by some kind of this pattern recognition receptor protection. Okay. So remember, this pattern recognition, those are all part of your innate immunity, right? Remember that they are coded by the DNA of the cell, and you don't touch that DNA. The way it is, that's what you're born with, that's what you're going to have. How many of those you have? Well, you can produce about 1,000 different types of, 1 second. No. You have hundreds, about 100 different type of PRRs, and you can recognize about 1,000 different PAMPs, okay, and DAMPs, PAMPs and DAMPs. So there are not many receptors here, about 100 or so, okay? Okay, so what else? You have this. So here, less than 100 different types of receptors, okay, variants. Okay, it comes in your DNA, so that means it's germline encoded in all the cells. Okay, all right. So when you compare here with your adaptive immune response, that's going to be totally different. Your adaptive receptors are going to be receptors on your B cells and on your T cells. And those are going to change. The DNA of those guys are going to change. It's a lot more complicated. Okay, so what is it that these patterns are recognizing, really? What is these patterns on the pathogen that has to be recognized? Well, first thing that you're going to recognize is what allows the microorganism to survive, meaning the genetic material. So you're going to recognize nucleic acids in your pathogen, but then you should start thinking that, wait a minute, my cells have nucleic acid. No, my cells have DNA and RNA. So now you're going to have some type of receptor that is going to recognize foreign DNA and foreign RNA and not your own, okay? So you have here certain patterns of nucleic acids that are specific for these microorganisms. For example, single-stranded RNA, that's a hallmark of viruses. You have double-stranded RNA, also hallmark of viruses. And then you have certain DNA that has like CPG islands, but we have CPG islands and now it's a problem. Oh no, but those guys in the pathogen, they're going to be a little bit different than your own DNA CPG islands. Those guys are not going to be methylated. Okay, so you're going to have a PRR that is going to recognize unmethylated CPG islands, right? So let's keep that in mind. Besides nucleic acids, your pattern recognition receptor is also going to help you identify proteins that don't belong to you. Those tend to be a bit easier, right, depending on the pathogen, of course. But for example, when you talk about pillin, that has to do with how bacteria do what? Anybody took micro? If I say pillin, what do you think about? Gee, you had a boring micro instructor. If I say pillin, you think bacterial? P-I-N? Sex! You think sex. You should think penis, but not quite. It's just sex. Bacterial sex, primitive, kind of boring. Okay, so that is very recognizable and you can attack it without killing yourself. Then you have flagellin. Those are proteins you find in flagella. Do bacteria have flagella? Some of them, they like to swim around. Some bacteria have one side flagella, so they swim like this. Some bacteria are confused. They have two sides flagella and they swim kind of like, you know the bacteria swim. I don't have to do the dance, right? But those are specific for bacteria, except for one cell in the human that has a flagella. Which one is it? Everybody should know that one. Sperm. The sperm, yes. You don't want to attack the sperm. All right, okay, so this one is attacking your bacteria, okay? Bacteria. And then you have other things that could be easily recognized, like cell walls, because we don't really have cell walls, right? So cell walls, lipids. Now let's pay attention to this, because the biggest cell wall you're going to find in gram-positive bacteria, and the not so big cell wall is going to be found in your gram-negative bacteria, right? Cell wall lipids, okay? So we're going to have to figure out which one is which, and you're going to have to know those, right? So if you look at LPS, that is lipopolysaccharide. You're going to find that in your gram-negative bacteria, okay? Then you look at lipoteichoic acid. That is characteristic of your positive, gram-positive bacteria. So lipids in your cell wall, that's important for your patterns receptors to recognize, okay? Because we don't have those in our cells. And then you're going to have certain recognition of carbohydrates, okay? And carbohydrates that, of course, are not really present in high numbers in your own cells because you don't want to kill your own cells. So what are those? You're going to have mannan and glucans, and those are very abundant. Oh, I think it works in bacteria and also in fungi. Okay, mannan especially for your fungi. All right. So these are all your pathogen things. Now remember, your PRRs also have to recognize damage stuff in your own cells. So important damage things that you should know. If your cell is under stress, it's going to produce stress-induced proteins. So those are patterns that also can be recognized. And if your cell is producing too many crystals, those also can trigger some recognition. And in this case here, look, you have accumulation of urate crystals, and that can cause this inflammatory reaction. It is called gout, okay? So your pattern recognition receptors are crucial on triggering this gout. Anybody had gout here before? Painful, very, very painful. Do you know where you usually get gout? Bless you. It's almost like it, but not quite. Okay, let's think about it. Gout is accumulation of crystals. Crystals are heavy. So if you're going through the circulation and the crystals have to accumulate somewhere, where do you think they're going to accumulate? On your feet somewhere, because they're heavy. They go down and they don't like to come up much. And they like to stay in a joint, okay, in a cartilage of some type. And specifically, they like to get stuck on your big toe cartilage, on the big joint in your cartilage, in the big toe. That's a very common place to get a gout inflammation, and it is so painful. It's like you can't even stand like the bed sheets touching your toe. It feels like fire. It's so painful. How do you get gout? If you are a carnivore, if you like to eat your meat or fish, you eat a lot of nucleic acids, right? Proteins that are filled with nucleic acids, red meat especially, and fish, salmon, okay? Breaking down of those purines in the nucleic acids can cause crystal formation if you have too much of it, okay? And that can cause gout. So a balanced diet with not too much, of these animal-based proteins. It's a good idea to not have gout. Okay, so gout used to be called the king's inflammation disease or something like that, because in the medieval times only the very rich people could afford the meat or the salmon or anything like that, right? But nowadays it's like we can all get it, right? Hopefully, I mean, if the grocery prices go down anytime soon. Anyway, so. All right, we're going to see into more details on the mechanism of gout, but keep in mind that what is causing all the inflammation is really cells in your innate immune system. All right, what else is important here? Oh, look at this. If you have nuclear proteins that wandered out of your nucleus and are outside of the cell and they're just like where they're not supposed to be, those can trigger inflammation. Okay, so you have some pattern recognition that can go and recognize, for example, histones that are not in the nucleus and they're floating around and they're not supposed to be floating around. So that can trigger inflammation. Okay, so these are ballpark pictures of what these things are recognizing. And now look what I said here. Okay, you have lipopolysaccharide in your gram-negative bacteria, right? And that we're going to see what is recognizing that. So remember something about gram-negative, something about gram-positive, something about fungus, and then something about viruses, because those are the major types of pathogens that you can have. Okay. All right, so here is that statement saying that your innate system is going to recognize things that are absolutely needed for your microbe to survive. So the microbe can't just start mutating things, trying to get rid of that thing that it needs to survive, because if it tries to, then it's going to die. Okay, so this is a very good strategy of recognizing things that are vital for the microbe survival. Like, for example, as we mentioned, double-stranded RNA for the virus, and LPS for your gram-negative, and lipoteichoic acid for your gram-positive. Okay, so your bacterial cell walls, gram-positive, gram-negative. Okay. All right, so where is it you're going to find those receptors? They're everywhere. Okay, they're inside of the cell. They are on the surface of the cell, and they are soluble outside. Okay, inside, outside, they're going to find them everywhere. All right, so let's see here again what is very, very important for that recognition. If you look at this bacterium right here, and you see lipoteichoic acid, you can guess that this thing is going to be a gram-positive. If you see lipopolysaccharide, that's going to be a gram-negative. But also in the cell wall, so this is just, either one or the other. In the cell wall, you're also going to have those sugars, peptidoglycan, okay? In the fungus, you're going to have mannan, okay? Typical for cell wall in the fungus, all right. All right, so remember that your PRRs, you have two types of PRRs. One of them that signals the cell to eat, right, the pathogen, and another one that is going to signal the cell to send signals out. So you have your phagocytic PRRs, and you have your signaling PRRs, okay? So here, let's look at what is happening to this poor bacterium that got recognized and eaten, okay? Once the PRR is recognized, again, this is not very depictive of what is happening because you need to have at least two of those patterns being recognized. And now it's going to be a bit more clear why that is the case, okay? So let's say you have two patterns recognized. This thing ate the bacterium. At first, you have this little vesicle that is called the phagosome. Then you're going to have lysosomes that are going to fuse to it and bring enzymes to kill the bacterium, okay? And you call this thing here your phagolysosome. So what is happening inside the phagolysosome? Two things can happen here. The first thing, when the lysosome just fuses with your phagosome, the environment inside of this phagolysosome has very much a neutral pH. Okay, so in this environment here, you have certain proteins that can start killing your phagocytosed thing. Okay, but at a neutral pH, you only have very few proteins that they can start acting. So you have some proteases and some hydrolases that are useful at this neutral pH, but that's not enough to kill the pathogen. Okay. Then what starts happening here is that you're going to have little pumps in this phagolysosome that is going to start making the inside of this phagolysosome is going to start making it acidic. So you're going to pump hydrogen inside of this place. So this place is going to get acidic. Once this place, the pH goes down, like less than 5, for example, okay, then you have other little granules that are going to get activated. Okay, so you have other enzymes that are going to get activated. So those enzymes are going to also help to start killing the pathogen. Okay, now, so you have the two processes going on here. Enzymes that are working at neutral pH, and then the other enzymes that start working at acidic pH. And still, with all of these enzymes getting activated, how much of the pathogen that you killed so far? Less than 2%. So this is not very effective, like at all. Okay, so you need to have a better strategy of killing your pathogen that got engulfed. Okay. So what do? This is when your innate cells, some of them, 2 in particular, they can go through this process of oxidative burst. You can also call it a respiratory burst. And when you talk about something respiratory, you should think about oxygen. Okay? Oxidative, oxygen, respiratory, oxygen. So what's going to happen here? You have 2 cells in particular that are very good at doing this. One is a macrophage, and the best one is really the neutrophil. Okay? So they eat up the pathogen and they lower the phagolysosome environment, and still the enzymes are not doing it. So now what's going to happen is that you're going to start producing nitric oxide, and you're going to use oxygen, of course, to produce nitric oxide, and you're going to use oxygen to produce very unstable reactive oxygen species. Okay, it's like ROS, reactive oxygen species. Those are your superoxide anions and also hydrogen peroxide. Do you guys have hydrogen peroxide at home? Like first aid kit? Yeah? Yeah, what does it do? Cleans the wound? I mean, water can do that. Yeah, but how? Oh, it's really toxic to the cells. The cells are going to burst with these reactive oxygen species. Okay? And this is your, actually hydrogen peroxide is light sensitive. So if you look at the bottle that you have your hydrogen peroxide, it's usually this dark brown bottle to protect it from degradation. Otherwise, it becomes water and then it's not effective, right? So these reactive oxygen species are very effective at killing the pathogens, but guess what? They also kill the cell that produced them. So this is a type of a kamikaze mission. Okay? The cell is going to die. All right. So these reactive oxygen species, they can bind and react to a lot of things, like cell walls, like proteins, like DNA. They will bind and react with whatever they can bind to. Okay? So they're very, very toxic. And they're also going to cause tissue damage. And this is another reason why after you have your innate immune reaction that killed the pathogen, you also have to go there and fix up the tissue because your cells damage your own tissue. Okay. All right. So how is this burst happening? Inside of your 2 cell types, remember the cell types? Macrophage and neutrophils. Remember those guys. Neutrophil is the best one. Okay. You're going to have oxygen, okay? You're going to have some floating amino acids like arginine, right? And you're going to have this guy here, look, NADPH. This is nucleic acid that is also an electron carrier. Anybody took biochemistry? No? Yes? Oh, who took biochemistry? Yeah, where do you find NADPH? Pentose phosphate pathway, that kind of thing, like sit away from your teacher, there's spit going over, pentose phosphate pathway. NADPH is a molecule that is very important for building up things, okay? But is also very important for producing reactive oxygen species in these neutrophils or macrophages, okay? So you need to have an NADPH. All right, not only you need NADPH and oxygen, the reactive oxygen species, can do without the oxygen, right? Not only you need that, but you need an enzyme to do the job. The enzyme, let's see what you call it, inducible nitric oxide synthase. Guess what the enzyme is making? Yes! It's making nitric oxide right there, okay? And it's inducible because you're just not going to be making it just because they want to. You have to be triggered if you have that phagocytosis and the phagolysosome going on, okay? Now, nitric oxide is very important, not for killing the pathogen, but is important for dilation of blood vessels. So let's keep that in the back of your head, okay? All right, and with that, you're also going to produce those reactive oxygen species. So let's look at this reaction here, okay? The phagolysosome, okay, when that thing starts to lower the pH, right, you're going to activate that enzyme, and you're going to also activate this NADPH oxidase. Okay, so here NADPH is going to use NADPH and is going to use oxygen. And what you're going to do, you're going to get an electron from NADPH. Remember, that thing is an electron carrier, and you're going to transfer it to oxygen. So you're going to make that very reactive oxygen species. Okay, now with a reactive oxygen species, what is going to happen to your NADP? That is going to lose that electron, also with a proton, right? And it's going to become NADP plus, okay? So NADPH donated that electron to make the reactive oxygen species, donated the proton to be floating around and still lowering the pH, and now you have those superoxide things floating around. Now, this peroxide ion, it is very, very reactive. That is going to kill your cell very fast, and you don't want very fast killing of everything. You want to have controlled killing. So besides that, you're also going to have an enzyme that is called superoxide dismutase. And what is that enzyme doing? It's going to unmake your superoxide. It's going to make it into something else. It's going to make it into hydrogen peroxide. Okay? So it gets this highly reactive superoxide and transforms it into hydrogen peroxide. Right? So the enzyme is doing what? Is using NADPH, is making reactive oxygen species, then you get your superoxide dismutase and you get hydrogen peroxide, which is still highly toxic, but it is more targeted for killing whatever is inside the phagolysosome first. Okay, so you got to know these reactions here and what the enzymes are doing. Very important for your oxidative burst. Okay, now look at this. If your body has a problem producing the enzyme that allows this first reaction to go through, then you're going to have a bigger problem with pathogens that are phagocytosed. Okay, what happens here is that if your body cannot produce this enzyme, NADPH oxidase, then you're deficient in the enzyme, and what is going to happen is that you're not going to have oxidative burst. Now remember that oxidative burst is the effective thing of killing your pathogen. What is the other way you can kill the pathogen in the phagolysosome? You have something else that kills the pathogen? The enzymes, right? But remember how effective those are? Less than 2%. So what's going to happen with all the 98% of phagocytosed pathogens if you don't have an ADPase oxidase? You're going to survive, okay? But your cells are trying really hard, your neutrophils are trying really hard to eat them and kill them, but they can't kill them effectively, but they're still trying to eat them, okay? So when you look at those cells under the microscope, you're going to see that the phagolysosomes are filled up with these pathogens that are not digested. And when you look at it, they look like little granules that are bigger than they were supposed to be, and still the thing is not dying. So you look at it and you go, oh, look at this. I see chronic granules or granulomatous disease. So this is a type of immune deficiency that you're born with, okay? Chronic granulomatous disease is a type of immune deficiency of your innate immune system, which can be quite serious because these people are susceptible with all types of infections, okay? They should be taken care of by your innate immune system, okay? All right. By the way, I like to ask questions about this because it shows your reasoning, right? So I like to put little scenarios on what happens if this person gets this and that. You look at the cells, you see this and that. So imagine what kind of questions I can ask based on this information, right? Okay. All right, so let's look more into your pattern recognition receptor, right? Remember, you have those signaling PRRs. We look at the phagocytic ones. So now looking at the signaling PRRs, they're going to produce those little molecules that are going to be secreted, dumped into the environment. And these molecules are going to start calling on their friends, other friends' cells, to come along and reinforce their attack, okay? Because they need help. Okay, why do they need help? Well, because whatever pathogen attacked the system, the pathogens usually replicate very quickly, and if you don't have enough cells to go down and kill them, then you know you're gonna lose the fight. So those cytokines are very, very important for communication between cells, and also to attract cells to the site of infection. Okay, so signaling PRRs. The most important ones are really the toll-like receptors, okay? How do they look like? Well, they're like little hooks like this on the surface of a membrane. So this is a membrane. So little hooks that goes through the membrane here, transmembrane little hooks, okay? But if you look at it, it is a single chain. As a single chain, they are not going to work. They're just going to be quiet there. Okay. And where are you going to find them? You find them in epithelial cells, endothelial cells, and all of your white blood cells. All of them. Okay? You're going to have TLRs. All right. So TLRs. What are the ones that you have to be familiar with? 1, 2, 4, 5, 6. Yeah, there are lots of them. So these guys, 1, 2, 4, 5, 6. They're going to find them on the cell surface. And they are going to be very good at binding to PAMPs. What kind of PAMPs? Peptidoglycans, lipopolysaccharide, and flagella, right? 1, 2, 4, 5, and 6. And then you have other ones. There are 3, 7, 8, and 9, okay? Those are going to be inside of the cells. Those are going to be very important to dealing with the stuff that gets through the membrane and gets inside of the cell. So those are going to be very important against viruses. Okay. Those TLRs, they are going to activate something that is inside of the cell, and that is going to be a transcription factor that is going to go all the way to the nucleus and tell your cell to start making proteins to kill the pathogen. Okay, so this is going to be activation of your NF-kappa B pathway. Okay, so TLRs are going to do that. Okay, besides upregulating anti-pathogen proteins, your NF-kappa B is going to increase the inflammation, and is also going to secrete some more of those pro-inflammatory cytokines. Now let's wake up here, because you absolutely have to know what are the most important triad of pro-inflammatory cytokines. One of them is interleukin-1 beta. Second one, TNF-alpha, that's called tumor necrosis factor alpha. Third one, interleukin-6. Those three are very, very important inflammatory cytokines, okay? And they're going to do all sorts of stuff. It's going to go to your liver and tell your liver to produce more of fighting enzymes. They're going to get to your brain and tell your hypothalamus to increase your body temperature. And that means you're going to have fever. And this is why I was so worried after my baby ate that piece of whatever that was, right? Is he going to get a fever? You know, is all this pathway going to be activated? Is he going to survive? I'm such a bad mother. You know, believe me, every time, I mean, whenever you decide to have a child, you always think you're a bad parent. But they survive somehow. All right. Okay, so I got a few more minutes here. Let's finish this slide here. All right, so you have intracellular type of defenses, okay, that is going to include secretion of antiviral proteins, and the first line of defense against viruses is really your interferon, okay? So you're going to secrete interferon alpha beta, okay? That's going to interfere with viral replication. All right, so let me see here. Yeah, we're almost close to the end. I'm going to stop in this slide here, and we'll pick up from this next time.